Hi, so now we're going to move into part two with a much more modern perspective on all of the ideas you've just seen about forms and patterns and archetypes. We're going to focus on the work of Michael Levin, who appears to be one of the leading, if not the leading person in this area. I wouldn't be surprised if he receives a Nobel Prize in biology sooner rather than later. So without further ado, let's just plug ahead a little bit and we'll see why we're saying this. Michael Levin, by the way, was born in Moscow and in the 1990 timeframe came to the United States. His father was a systems computer programmer, and so he has a very strong computer background, but was always fascinated with biology. So let's just take a look at where Michael is coming from on all of this here. For over 100 years, biologists have been working to fully understand biology at the level of chemistry. In other words, at a very low level, working up, in other words, to unite biology with chemistry. And this, many call this the reductive approach, uh, which is inspired by the unification of chemistry with physics, with the periodic table and quantum mechanics. And all of this meant, well, if it works so great for chemistry, working with physics, let's do the same thing for biology. And the major player in this is the gene and things like DNA and RNA and genomics are the very bottom-up approach you'll see to this. This is, if in the computer sense, this is very much like dealing with the hardware. There's very little discussion here of the software, of what makes things life. That's what separates biology from chemistry in many ways. So the gene-linked phenotype to the molecules of chemistry and to the more abstract and promising world of information. But again, it's still bottom up. Now, this is not to disparage what's been done. There have been many great successes and new understandings in treatment of cancer, using what's called gene therapy for things like sickle cell disease, but there have also been many failures. Uh, there are many estimates that suggest we might be able to treat 1% of all the diseases that we find using this approach. And that's not too good a target to come up with. The drugs that have been developed flop, they're not approved. The diagnostics fall far short. We've seen the disclaimers again and again, the side effects of these drugs. And the net of it is that much about our body remains shrouded in mystery. And we have heard that if DNA sequencing, which is the way we characterize the genes of the genomes, was much cheaper and we just did more of it, then we could solve these questions. Well, right now, to take some DNA, there's 50 trillion cells in your body. Each of them has two strands of DNA. They're roughly 2 meters long, 6 feet. So they're folded up into one another in an incredible folding pattern. And we can now sequence these things for on the order of $100 or less. So we've reached that. Well, is nirvana here? I don't think so. So Michael Levin is leading a completely different approach, the one that we built upon in our first part of this talk. And this is a non-reductionist approach where they're looking for answers beyond the genome. Michael is at Tufts University right now, and he calls himself a synthetic biologist. But he directs a staff of roughly 50 graduate students, and he also has ties to uh, Harvard. But he focuses on the innate intelligence of tissues and organs. So just right away, this leaves you, what innate intelligence? What in the world is going on here? He argues that the networks of cells are goal-directed. So they achieve their goals through the medium of bioelectricity. This is the electricity that is around the human body, and it's biological in nature. So this somehow is some sort of brain that connects everything through this goal-oriented activity. And he knows that terms like intelligence and goal are taboo, And so he insists on fundamental basic definitions for these terms. The idea is to go in and reprogram the goals of a group of cells and let them do the work of reversing whatever the disease is. So if you talk about gene therapy, Mike says he can imagine biological machines that are already in our body, finding and repairing damaged DNA, tissues, and organs. That's his view of gene therapy. That's very different than if you talk to many other people in mainstream biology. He's already published over 300 publications. Yeah, there are these two websites here that you can go to and see some idea of what he's doing. There's an incredible number of YouTube videos, and we're going to look at one of them right now to give you some idea. But this is all building on the ideas we talked about before of patterns, archetypes, forms, resonances, and this is his take on this for biology. Marjorie, anything?
Oh, well, thank you for asking. No, not yet. I hope people have seen part 1 before they tune in to part 2. That's all I'll say. It'll make a lot more sense if you see
Sure.
part 1 first.
Sure. Okay. So here we go. We're going to look at a video here. I'm going to make sure, let's see if this thing is cranked up. And okay, we're off to the races.
Sitting down with Michael Levin, who is working on a paradigm shift in medicine and biology.
You can inject human oncogenes into a tadpole and they will make tumors. And if you do that, but you force the cells into proper electrical state with communicating with their neighbors, you prevent tumorigenesis, even though the oncogene is blazingly strongly expressed.
Cancer is the second leading cause of death globally, and we fight the disease primarily with chemotherapy, radiation, or surgery. But what if there would be a way to tell cancer cells to stop growing through the use of bioelectricity?
What you're doing is you're forcing that cell to stay connected to their neighbors. And when you have a mind meld with a large collective that all says, we are building a nice kidney, we are building a skin, it's really hard for that cell to have an independent thought of, you know what, my goal is to proliferate as much as I can and crawl away wherever life is good, right? Metastasis.
Okay, so what we want in our bodies is a mind melt of cells.
Confidence isn't something you have, it's something you do. As behavior researchers,
that cells forget about their own little ego and start identifying with a larger collective of the organ. And the way cells do that is through bioelectricity. It is what Michael Levin calls a cognitive glue.
What's actually important about bioelectricity is that it functions as a cognitive glue. And what I mean by that is, you know, think of how it works in the brain. We are a giant collection of an enormous collection of individual neurons. And yet those neurons come together in some way to give rise to a new individual that has goals, preferences, memories, and capacities that none of the individual neurons have.
So basically, we are a collective intelligence of cells held together by bioelectricity. Now, what cancer cells do is they disconnect.
The first thing that happens when oncogenes act, first thing they do is they cause electrical disconnection from their neighbors. And as soon as you disconnect it, well, now you're an amoeba again. And as far as you're concerned, the rest of the body is just external environment. So that border between self and world can shrink and grow.
Now, what's groundbreaking about the work of Michael Levin and his lab is that they found a way to tell cancer cells in frogs to start cooperating again.
And we've done this in the frog model. We've already done this. So this stuff is not just wishful philosophical thinking, and no, it has very practical implications that if you follow through, you discover therapeutics.
But things get even more exciting, because in bioelectric fields, Michael Levin and his lab found blueprints of organisms.
So we have now tools where you can literally see, for example, the electric face. There's pre-pattern that tells you where should the eye go, how many should there be, where should the mouth be, how many heads should a planarian have. These are states of distributed bioelectrical circuits that are now visible. You can see them. Rather than being able to see them, you can change them.
In this planaria, so-called voltage patterns were altered. telling it to grow two heads.
We don't touch the hardware. We don't change the DNA. We don't tell any of the stem cells what to do. All of that is taken care of by the system. All we do is reset the goals. You get to make complex outcomes that nobody could make before because the micromanagement is too hard.
And this is where we hit on a paradigm shift, because normally in biology and medicine, we focus on genes and molecules and managing those. You could say we focus on managing the hardware of cells, whereas Michael Levin focuses on the software.
All there is molecular reactions. So if you want an extra eye or a worm with two heads, just manage the molecular reactions. That's all you get. That's the conventional view. And the implications of that conventional view is that you really need to micromanage all of those chemical reactions to get what you want. Now, that's incredibly difficult because the mapping backwards, if I know what I want, well, what should every gene be doing? What should every cell be doing? What should every molecule be doing? That's really hard.
In developmental biology, there's the study of morphogenesis, the origin of forms, the way how cells organize themselves into shapes and tissues. Now, many aspects of morphogenesis still remain a mystery to us.
No individual cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely does. And that's how you get to things like a salamander limb, where you can amputate anywhere along the length, and the cells will build exactly the right thing. And then the most amazing part is they stop when they're done.
Pondering upon Levin's work, I asked him where he thinks that the memory of forms in nature is stored.
Plato and Pythagoras had this idea that there is a Platonic space of these kinds of things, of these forms. And I think we need to take this seriously, in that I think one thing that evolution is doing is searching through pointers into that space. So I think evolution is not really searching the horrible, complex, rugged space of microstates of all the hardware, the phenotypic space. I think evolution is actually looking for pointers into this incredible space of forms that already pre-exist. Even bacterial biofilms, the very first kind of attempts at multicellularity, at a kind of weak form of multicellularity, were already using electrical network. Even individual bacteria living in a network form a kind of collective that does better than the individuals do, because it uses bioelectricity to integrate information across space and time.
A wonderful practical implication of Michael Levin's work could be that in the future we can also, in humans, tell cancer cells to stop, which would be fantastic. But what I find just as important is a deep philosophical lesson his work teaches us, namely that we should do away with all categorical thinking around consciousness and intelligence, because it's all a matter of scaling, according to Levin, and we ourselves could be part of a larger intelligence without knowing it.
I certainly think it's possible. Absolutely, I think it's possible. But I think it's very important that we don't fall into the trap of, some people say, that's ridiculous, that's a misuse of the terms. And other people say, oh, absolutely, the whole universe is one giant mind, like, okay, both of those are hypotheses. You cannot decide between those just by sitting in your armchair and deciding which one feels good to you. You need to do experiments. And obviously on the higher level, on the level of the universe, the level of the solar system, I can think of how to do those experiments, but they're not practical. I wrote a little, dialogue at one point between two neurons in a brain, and one neuron is kind of like the hard-headed materialist, and he says, "Look, we live in a cold, unfeeling universe. There's no pattern. Nobody cares about us. It's just each one of us is alone." And the other one's a little bit mystical, and he says, "I don't know. I have this feeling that we're part of some system that actually, the universe actually learns and wants things and does." And the person's, you're crazy, And he said, "No, I don't know. There's like these patterns of, of propagate, like back propagating error and whatever. And sometimes, the universe punishes me and whatever. But I feel like there's a purpose behind it." And he says, "That's crazy." So right. So from the perspective of in that system, the mystical neuron is correct.
Welcome to the Essentia Foundation's YouTube channel. If you're new to our channel, may I ask you to like and subscribe, as it really helps us to grow and make more content like this. Now, without further ado, here is my full conversation with Michael Levin.
You've been able to tell organisms, Panaria, and to grow two heads, right? By cracking the code. And not only did they listen to that by growing two heads, but the next generation of planaria also did that. And there just, to me, doesn't seem to be a very good explanation for that if we are in sort of this old paradigm of evolution, life, and biochemistry. So my sort of main question to you as an opening question is, what sort of new paradigm are you working on, or what do you think will be the next paradigm to make sense of what you and your colleagues at your lab are doing?
I think it's important to say that any phenomenon, including the two-head thing and anything else, if after it's done, after somebody shows how to do it, you can always tell a standard molecular story. That is, after the fact, you can always zoom down to the molecular events and say that, oh, well, there's nothing special here. It's just molecules doing what they do. It's normal laws of chemistry. Of course, everything works. And so we have to think about the difference between telling causal stories backwards, trying to explain what happened, versus the kinds of paradigms that suggest new experiments and take science forward. So this gets into this issue of for example, reductionism and determinism and things like that. A very simple example is if there's a chess game that was played, and you can look backwards, and a reductionist can say, look, I tracked all the electrons and all the protons, and everything worked exactly to the laws of physics. It was completely determined. There's nothing surprising here. And that's true. But how much is that story going to help you play the next chess game? Right? Not at all. These are, looking backwards, you can always tell a conventional story. And sometimes, especially with our work, you would have to put in some epicycles, so to speak. You would have to, put in some extra bells and whistles to make it fit the conventional paradigm. But you always can. And so if, when somebody says, look, this is what they've shown, somebody else says, oh, I'm sure there's a molecular explanation for it. Of course, there's a molecular explanation for it. There's always a reductive explanation for it afterwards. The real question is, what was the paradigm that got you to do the experiment in the first place, and why wasn't it done before? And so, the first two-headed worms, I mean, there are many ways to make two-headed worms, and the first two-headed worms were seen at the turn of the last century, so 1903 or so. The question, though, is, since between then and 2009, when we did it, why did nobody recut them? To my knowledge, nobody ever recut them. And the reason you wouldn't recut them is because if you have a standard belief that your genome is what determines your morphology, and the genome hadn't been touched, well, then why would you do it? Of course, you have an expectation. Of course, they will go back to being one-headed once you cut off the ectopic head. And our paradigm predicted the opposite, because we were thinking of the bioelectric circuits that store this information as a kind of memory. And the most obvious thing about memory is that it keeps state, either temporarily or permanently, but it keeps state. So I think that's important, right? It's not just the phenotype. I mean, anybody can get a double head. There's many ways to get a double head. The new, so just to answer your specific question, the new I think what we have here as a new paradigm is this idea that everything exists on a spectrum of cognition, and that you cannot say where something is on that spectrum from philosophical commitments. You have to do experiments. So when we ask what is the correct way to communicate or to understand cells, to communicate with or to understand cells.
Ready to join the booming cruise industry? Become an Expedia Cruises franchisee. You'll be part of the Expedia.
You don't know that the standard machine paradigm, meaning with the paradigm for simple machines, this kind of bottom-up emergence, complexity, dynamical systems theory, you don't know that those are your best tools until you try. It could well be, and I think it is the case, that a lot of these systems, subcellular molecular networks, cells, tissues, organs, are actually much further along the spectrum. And that means that what you're really doing, if you want to predict and control and build new things, is you have to use tools from behavior, from the behavioral sciences. And so now this is an interesting thing. I mean, this in drawing this spectrum and in being really open to what kind of systems might be anywhere on the spectrum, this goes against both of the two dominant paradigms, because you've got the kind of the molecular biology paradigm, which says, look, it's crazy to it's a category mistake to think that these kind of behavioral / mental kind of primitives, goals and memories and things like that, it's crazy to attribute those to these simple systems, and that what you need to do is everything needs to be cashed out in the laws of chemistry because these are simple machines. Okay. Then of course there's the organicist position, which has been fighting this view for probably centuries, which says that no, look, living systems are special. They cannot be treated as these simple machines. They are essentially different. There's something categorically different about living systems, and maybe living systems are cognitive systems, but the point is these are not machines. Okay, this, and so the perspective that I bring goes counter both of those, because I'm saying, no, actually, there is no categorical difference. What there is a spectrum of scaling, and that even the simplest things have certain properties that might be usefully exploited by a cognitive approach, and that all what we need are not stories of categorical differences and emergence. What we need are stories of scaling. We need to understand how the very simple competencies of even simple molecular networks are capable of learning and things like that. And so what we really need to understand is how cognition scales up. And so that's the idea that we need to be free of these old categories, right? That's another thing, when people say it's a category mistake. Well, categories aren't, fixed by philosophy and then we hold on to them. I mean, it used to be a categorical mistake a category error to think that the same laws could apply to the motion of physical objects here on Earth and motions up in the heavens, right? That was at one point that was considered a terrible category error. So categories need to shift. The science needs to tell us what the categories are, not the other way around. And I think that both of these stories are now crumbling. These categorical stories are crumbling.
Even point to sort of the mind-body story, sort of that dichotomy is also sort of not very useful, right? And a question I have, Michael, but this is again speaking in dichotomies and in sort of categories, but just to help viewers understand, we usually have sort of this hardware, software sort of view on things in our era of computing. It's just the metaphors we like to use, right? And then sort of your DNA would be somehow the software, and other proteins within the cell would be the hardware, right? And you also sort of crumble that whole distinction, right? And is it right that, and I was thinking, would one way of looking at it be that we have this analog way of thinking, in the sense that, I mean, in the old days, the analog computers, the algorithm was in the hardware, the configuration of the hardware, right? So in this, compared to the cell, it's sort of the how the proteins are configured. It's a hardware story, and you sort of have discovered, no, there is software, but it's not sort of on that hardware level. So it's a different paradigm of computing within the cell. Is that a way of looking at it? Is that a useful way of thinking?
Yeah, I mean, I think the first thing to nail down is this idea that all of these things that we talk about, hardware, software, instructions, representations, Turing machines, all of this stuff, these are not claims about the real system. People always say, but some people say that living things are not computers, they're not machines, right? Because nothing is anything. All of these things are frames that we take, when you adopt a computationalist perspective or some other perspective, you're not really saying that's what this thing really is. Okay, nothing really is anything. What you're saying is, this is the frame that I'm going to take in interacting with that system. Let's see how far it gets us. So that's all we're really arguing about. And the great thing about that is that multiple observers can have, and so this is something that Josh Bongard and I developed, which is a perspective called polycomputing, which is this idea that the same physical event can be seen as wildly different computations by different observers who interpret the same exact physical event in different ways. And so multiple observers can be looking at something and say, oh, I see this is clearly a Turing machine, and somebody else says, well, actually, I see it differently, and because of that, I'm able to do something else that you're not able to do. This is fundamentally important also in medicine. You want your orthopedic surgeon to think that you're a mechanical machine, right? If you have an orthopedic surgeon that doesn't think that your body is a mechanical machine, he's not going to get very far. But you don't want your psychotherapist to think that you're a mechanical machine. And so depending on what you're trying to do, different frames are useful. So the molecular biologists absolutely have useful have gotten useful mileage out of this idea that the DNA is the software. Yes, you can do certain things that way, but that's certainly not the only perspective. And there's another very useful perspective in which what the DNA does is simply define the hardware of the cell. So the DNA tells you which proteins, which ion channels, which promoters you're going to have, which determines that those determine the connection between the biochemical reactions that happen in the cell, and so on. That part's the hardware. And now you can ask, okay, so what is the physiological software that runs on it? What is the what's the plasticity of it? How do you communicate with it? What is it paying attention to? You know, what are the cognitive competencies of the software? And I also want to be really clear that by saying hardware and software, I absolutely don't mean that living things are well captured by the current architectures that we have. You know, this is I do not hold to a computationalist perspective where I think everything is a computation and that's all that there is. I do think it's a useful perspective in some cases, but it certainly doesn't capture everything that we need to do. And also, I think that people are often too hyper focused on the kind of the modern you know, whatever the current embodiments of these things. So people are thinking about, you know, current language models, and they say AI can or can't do this, and what they have in mind are these language models. Well, that's just, I mean, that's a tiny sliver of the space of possibilities in computer science. And the same thing with the idea of software and these architectures. You know, of course, living things are not a von Neumann computer, but there are interesting aspects of the software paradigm that are really, I think, useful in biology. For example, people who program have an interesting dualism that they buy into, which means that they have this paradigm that your algorithm determines what the computer does. It makes the electrons dance. Now you can imagine, and I wrote this you know, this little just for a joke, I wrote this little dialogue on my blog where this candidate comes to a software company for a job. You know, he's interviewing for a job, and the guy says, you know, you're going to code up some cool stuff for us. And he says, look, the computer is a physical device. It obeys the laws of physics. What's this magical algorithm that you think is gonna push around the atoms? No, they do what they do. You know, and so right. So it's not that that's wrong, but if you have that perspective, are you going to code anything useful? You're not, right? And so you need to really believe that these higher levels have a causal functionality to them. Otherwise, nothing happens. And I think, you know, people very naturally do believe that software actually does in some important way make the electrons dance, right? So that is really critical also in the kinds of things that we work on. And, you know, this idea that you can't just be stuck at the lowest level.
And just to, this is all just very fascinating stuff, but to reflect on sort of the mind, body, and also on this hardware software, though it is sort of a limiting metaphor, I know that, but how important you think are these bioelectric fields within, for instance, our own body? To what degree sort of is my makeup done by just genes and sort of totally explainable from a sort of molecular way of looking at it? And to what degree do we have to sort of invoke the stuff you are working on? Is there sort of something you can say about that for people who are new to this?
Sure. Yeah. Again, keep in mind that the idea isn't just to explain. Looking backwards, you can always explain things at the lowest level. In fact, sometimes when I discuss this with people, and I say, look, reductionism, you know, you want to reduce it to the lower level, and then you could say, so you want to be talking about quantum foam, right? I mean, reduce everything to quantum foam, and they say, no, that's crazy, you know, chemistry, not okay. So that tells you right there, you're not really trying to reduce. You've picked a level, and you sort of stay with it, right? So to me, the idea isn't to explain, because you can often explain things at this level after the fact. The idea is looking forward. So the idea is, okay, what insight have we gained to enable us to do the next great thing, which means new experiments, new research program, new biomedical treatments, new synthetic bioengineering to improve, you know, everyone's life. Which of these paradigms helps you make the next great discovery, right? And so I think that what's really important here is that if you do have this paradigm that it's the genetics that determine everything, and I mean, there are many people besides me. It's certainly not the only person to be saying that you know, the genetics is not a hard determinist kind of thing. I mean, many people saying that. But what I think is interesting and different about our work is that if you if you think about evolution as not producing solutions to specific problems, which is the typical way of thinking about genetic search, is that you're going to find the genome that produces something that it has the highest fitness in the environment, and that's it. You're looking for a solution to a specific fitness function. What I think evolution is actually doing is developing problem-solving agents. And I don't just mean clever animals that do things in three-dimensional space. I mean molecular networks that can learn cells and tissues that solve problems in physiological space and anatomical space and so on. And so if you have this idea that what comes out of evolution are these problem-solving agents with competencies, with certain degrees of intelligence that is up to us to discover, that opens up a whole new frontier of research programs around how do we detect unconventional intelligence in operating in different problem spaces? How do we communicate with it in ways that are effective to change their behavior, right? So for biomedical purposes and so on. And of course, it will also have ethical implications in terms of making us much more open-minded about what kinds of things deserve care and compassion and so on. That's the idea that through the bioelectricity isn't magic, right? Okay? There's probably, my guess is that throughout the universe, there's lots of different.
Political corruption is why nothing works for ordinary people. It's why prescriptions cost a fortune.
Physical modalities that can serve as this kind of cognitive glue that I think what's important about bioelectricity here on Earth. But here, what I think evolution has done.
That's how you call it, Michael? Cognitive glue? Sort of cognitive glue is how you bioelectricity is sort of cognitive glue.
It's a kind of cognitive glue. And what I mean by that is that I think it would be a real mistake to see the bioelectricity as just another piece of chemistry and physics that you need to add to what we already know about, methylation and all these other chemical gradients and all this stuff. Okay, fine. So here are some voltage gradients. I think you could do that, and some people do that, but I think that's a big mistake because what's actually important about bioelectricity is that it functions as a cognitive glue. And what I mean by that is, think of how it works in the brain. We are a giant collection of an enormous collection of individual neurons, and yet those neurons come together in some way to give rise to a new individual that has goals, preferences, memories, and capacities that none of the individual neurons have. So we exist because we as human minds exist because the individual neurons and other cells in our brain are tied together via some interaction policies and some mechanisms that underlie those policies that enable them to function as a whole, as a collective that is more than the sum of its parts. That's the cognitive glue. Now, in the brain, it's not controversial. We know what it is. In the brain, the cognitive glue is the electrophysiology that runs the electrical networks in our brain. So my point is that evolution did not wait until brains evolved to make use of this. This is extremely ancient. And even bacterial biofilms, the very first kind of attempts at multicellularity, at a kind of weak form of multicellularity, were already using electrical networks. And there's this very nice data on this from Gorol-Suell and other people showing that even individual bacteria living in a network form a kind of collective that does better than the individuals do because it uses bioelectricity to integrate information across space and time. So they use information, they use bioelectricity to share nutrient information across the colony, and they use it to organize feeding cycles as a kind of biorhythm inside the.
That's amazing.
Right.
That's amazing.
Yeah. Oh,
so
no,
we knew already, of course, about molecular signaling, but that's sort of the hard way of secreting molecules that sort of inform the sort of a colony of bacteria, right? But this is these fields. I'm just so, and this is really a lay question, but I mean, what is needed? What's the substrate needed for these fields? Sort of ions, or what is needed sort of create these fields?
Yeah. So again, I'll point out that the cognitive glue function could, in theory, be implemented by many other mechanisms. Maybe out there in the wide universe there are other ways to do it. Probably there is. But what I like about bioelectricity is that it shows us one clear example of how it happens. And I'll tell you how I see this happening. So a single let's just talk about single cells. A single cell, and by the way, I don't believe that single cells are the origin of cognition. I think it goes well below that, but let's just
serious. Okay, let's go into that as well.
We can get into that. yeah, no, I think I think cells are already, you know, kind of high on that spectrum. So here's how it works. If you have a single cell, within the cell membrane, you will have these little proteins called ion channels. And these proteins have the ability to move charged molecules, you know, so potassium, sodium, things like that, in and out of the cell. That gives rise to a voltage gradient between the inside and the outside of the cell. Some of those channels, this is quite clever. Evolution discovered that some of those channels can themselves be voltage sensitive, which means that what you really have is a voltage gated current conductance. That's a transistor. That's another way of saying transistor. And so that gives the cell something very interesting, which is a kind of memory or historicity, because if something happens and there's a particular voltage state, that changes the channel, which then changes the voltage state, which changes the channel, right? And so you have this feedback loop that can hold memories and it can make decisions and so on. It's already even right there alone with that one ion channel protein, you have some very interesting you know, atoms, so to speak, of cognition already right there. But when you put cells into networks, what happens is that the electrical properties of one cell can influence the electrical properties of another cell, just like in the brain, and the whole thing propagates. You know, you have information flow throughout the network. Now, the cognitive glue portion comes in the following way. One way that cells can connect, can pass information, is to send each other chemical signals. Now, if I secrete, so you and I are cells, I secrete a chemical signal, it floats over to you, you receive it with a receptor. It's very easy for you to recognize that it came from outside. And so, you know, it's a signal that came from outside, and you can ignore it, you can pay attention to it, you can interpret it in various ways, you can believe it, not believe it, whatever. But evolution found something very cool, which is called a gap junction. And a gap junction is a set of proteins on either side of two adjacent cells that they make a little conduit. You can imagine two submarines docking together via a little hatch. And so what happens when the gap junctions open between cells is that small molecules freely pass from one to the other.
Hey, Tone, why should people come to the AI Advantage Summit November 6th, 7th, and 8th?
Okay. Now this makes for a very very interesting thing, because let's say you have two cells. One cell gets poked. There's a damage event. One cell gets poked with something. There's a calcium spike, you know, that serves as a memory of the damage. That calcium spike now propagates to the next cell. Now the next cell can't tell that memory of damage belongs to somebody else, because a calcium spike is a calcium spike. It does not have metadata on it that say whose memory this is. So if you have a bunch of cells that are connected with gap junctions, what you find is that they have a really hard time separating which memories are yours and which memories are mine. It means that we are now in a mind meld that makes it really difficult, not impossible, but really difficult for us to have our own independent thoughts, because we are now sharing them. It's literally this kind of crazy mind meld. So the way the cognitive glue works, a few things happen. First of all, it's a mind meld with respect to a lot of different memories. That's the first thing. So now it's not me and you, now it's just we, as far as memories that define a cognitive agent. So you can already see how the the scaling is starting to happen. The second thing is that because lots of cells are joining together, their physical area of concern becomes bigger, so that now things that happen over here matter to me over here, right? And because signals take time to pass through the gap junctions, now even the temporal aspect is thicker, because now there's a, you know, there's a now moment, which is for the collective, which has a minimum width to it, right? Which is certainly the case in neural systems as well. Also, there's the notion of stress propagation. So if we think about stress as just the distance from the target of some homeostatic.
This is still very fast, right, Michael? If you talk about this temporal window to process sort of this field among cells, that's still very fast, right? Just electronics.
Minutes.
Oh, minutes. It can take a while. All right.
Minutes, because well, I mean, so one thing you can do is you can take any neuroscience paper and just do a simple find replace. You take anytime it says a neuron, you just say cell, and anytime it says millisecond, you say minutes. And then you got yourself a nice developmental biology paper.
All
because
right.
what evolution did is just turn the same, it just pivoted the exact same mechanisms from navigating anatomical space to navigating three-dimensional space. And then it had to crank up time because now you have muscles and nerves, you have to run around and not get eaten. So you know, developmental processes are slow. You have weeks or months to form a certain embryo or to regenerate or whatever. You have time. So all of that stuff is slower. So what's happening here is that you have your cell sheet connected by gap junctions. Now imagine if all of the cells are trying to maintain some kind of homeostatic state, let's say pH or hunger level or something, right? There's a target set point and they're trying to maintain it in a cycle. Well, stress is basically the measure of error, how far away from your goal you are, right? If all you do is keep your own stress, then everybody has their own little battle going on and everybody's doing their best, but it's just a collection of parts. But now imagine that you're starting to leak stress. You're starting to leak stress through gap junctions, through other mechanisms externally, whatever. If I'm sitting next to you and you are super stressed out and you're leaking your stress, now I'm a little bit stressed. And what happens is that in order for me to reduce my stress, one of the things I might want to do is help you, not because I'm altruistic, but because you're stressing me out. And if I reduce yours, so what's happening here is but this kind of stress sharing means that your problems become my problems, and things that are happening over there that are making some sort of a mess over here are now suddenly my concern. So what it does is what these kind of mechanisms do is they enlarge the cognitive light cone. The cognitive light cone is the size of the space and time bubble that you care about. What are you actually actively working to fix? And as it grows, your area of concern become bigger than a single cell. It's now the whole thing. Now having done all of that, you now realize that these gap junctions are not just empty pores. So they actually have state to them, meaning they can open and close based on things that happen. And now you have a fairly sophisticated set of connections. And the collective, the whole tissue collective has more computational capacity than any of the individual cells, meaning they can remember more things. So now what you've got a collective, whereas before it was every cell working for itself to optimize metabolic state, so you're working in metabolic space, maybe in transcriptional space, things like that. All of a sudden now you've got a collection of cells that has a much larger memory and computational capacity, and it cares about large scale things like maybe curvature, right? The overall curvature stress, or maybe other aspect of the size of the collective. Or now you've got a larger collective that can operate in anatomical space. And so this is how intelligence levels up. No individual cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely does. And that's how you get to things like a salamander limb, where you cut, you can amputate anywhere along the length, and the cells will build exactly the right thing. And then the most amazing part is they stop when they're done, because that error, that delta to the final pattern, has been reduced to acceptable levels. But that the pattern the set point that they're trying to recreate was something that no individual cell could do. And it's not in the genome. None of that stuff is in the genome. What's in the genome is proteins.
Where is it? Because one way to look at, when you say that's the amazing part, when it knows where to stop, right? When my cells know the body is sort of complete. And you also talk about, you even sometimes in your writing use the word story, right? There's a story, that sort of collective story of what the correct outcome looks like. Yeah. Where is that story stored or written? Because it seems to be accessed anew when sort of we have a new organism. I mean, this is what truly puzzles me.
Yeah. Okay. Well, the first thing I'll say is that if we really knew the answers to all those questions, we would be, the world would be unrecognizable. So I'm not going to claim to give you the final answer, but I can tell you what we know now. There are two questions here that you're really asking. Where is it? Where and how is it stored? And where does it come from? And what happens in new organisms? Okay, those are two very related but quite different questions. On the first one, we kind of actually know. We've developed tools to see it. So we can read and write, and to some extent, people have, in the biochemical world, people have talked about chemical pre-patterns and morphogen gradients. I mean, those are all conventional stories. We've added to this idea that we can actually see these bioelectrical patterns. So we have tools, much like neuroscientists do in the brain, to read memories out of the brain, right? So people are working on that. So we have now tools where you can literally see, for example, the electric face, this pre-pattern that tells you where should the eye go, how many should there be, where should the mouth be, how many heads should a planarian have. These are states of distributed bioelectrical circuits that are now visible. You can see them, and better than being able to see them, you can change them. That's what makes it clear that they really exist. Because another way of saying it would be, to go back to your original question, another way of saying it would be, forget all this kind of cognitive talk. all there is molecular reactions. So if you want an extra eye or a worm with 2 heads, just manage the electrical reactions. That's all you get, the molecular reactions. That's all you get. That's the conventional view. And the implications of that conventional view is that you really need to micromanage all of those chemical reactions to get what you want. Now, that's incredibly difficult because the mapping backwards, if I know what I want, well, what should every gene be doing? What should every cell be doing? What should every molecule be doing? That's really hard. But my claim is the reason that I think my claim about goal directedness and intelligence in these tissues is true is not just because we see these patterns, because you can also explain them at the molecular level. The reason is that by targeting those patterns and interventions, by using the techniques of changing memories, you get to make complex outcomes that nobody could make before because the micromanagement is too hard, right? So think about a simple neuroscience example of this is you have a rat and you want the rat to do a circus trick, sit on a little bicycle or something, and somebody says, okay, I can see this thing is a deterministic molecular system. So here's what we're going to do. We're going to find out all the different muscle motions that need to happen to make the body do it. Then we're going to track back to all the nerves that need to be activated. Then we're going to track it back all the way up into the brain. And then we're going to go through, and then I'm going to tell you exactly which pixels on the retina you need to activate with stimuli to make it do the thing. Now, in theory, that's possible. In reality, the sun will burn out before you figure out any of that. But someone who understands what the rat really is can say, no, you don't need to do any of that. You train the rat. What do you mean you train the rat? Top-down control. You collaborate with the rat. You incentivize the rat by and what's magic about that is that it then pushes that view of it, that top-down control pushes all the complexity onto the system itself. You don't need to figure out which neurons need to fire. The rat's already very good at knowing what needs to be done given certain top-level controls. It's a communication problem. It's not a micromanagement problem. So the same thing is going on here. These patterns, we know this is a goal-directed phenomenon because we change the goals. We don't touch the hardware. We don't change the DNA. We don't tell any of the stem cells what to do. All of that is taken care of by the system. All we do is reset the goals. That's the beauty of goal-driven systems, is that you can communicate with them. You can reset the goals. Maybe they learn, some cells and tissues learn. So we know where the information is stored. Okay, at least partially it's stored. It's represented by a distribution of voltage gradients across the tissue, and those gradients serve as the reference point for what to build and when to stop. Okay,
Michael, how robust is are these states, and how much of the organism has to stay intact for this information to sort of just be accessible again?
Yeah,
in the case of planarias, it seems that sort of just cutting off one head sort of didn't sort of the information was still there to grow know again it should grow 2 heads, right?
Yeah. A planarian can be cut into 275 pieces. So in planaria, it is incredibly holographic. The circuit rescales. So the interesting thing about that circuit is when you cut it into pieces, the circuit rescales. And so, in other organisms, it's not quite that robust, but still, even mammalian embryos, including humans, in the early stages, you can cut them into pieces, and you get monozygotic twins and triplets and quadruplets. You don't get half bodies, at least at early stages. So there is a lot of rescaling of this kind of thing going on. The second question you asked, though, is much harder and much deeper, which is where do they come from in the first place? And so let's just look at that for a second. Where do they come from, and what happens in new ones? Overall, the question of where things come from needs to be treated differently than a lot of the other questions we deal with, because and mathematicians have been on top of this for a long time, starting with Plato and Pythagoras, and probably long before that, which is that there are many things in the world that are kind of free lunches in the physics sense that evolution takes advantage of. For example, just this is a stupid example. Let's say in a certain world, it's very simple. There's an evolution going on, and the most fit thing in this world is a certain kind of triangle, certain shape of triangle. So evolution goes and does lots of generations, and it gets the first angle, and then lots more generations go by and it gets the second angle. Now here's the magic part. You don't need to do any more. You know what the third angle is. Yeah, you get it for free. Now, isn't that amazing? Isn't it? This is not philosophical fluff. This is incredibly practical. Evolution gets to save 1/3 of its time here because of this incredible free gift from geometry, the fact that once you know two angles in flat space, you know the third. So now there's tons of this stuff, tons of it. You know, when you make a voltage sensitive gap junction ion channel rather, you have a transistor. When you have a couple of transistors, you have a logic gate. When you have logic gates, you have truth tables, and you can build, you know, whatever functions you want. Do you need to evolve the elements of that truth table? No, you get it for free. Do you need to evolve the fact that the NAND, the not AND gate is universal, it's special? No, you get that for free. Where does it come from, right? Where do these things live? The, you know, the rules of, the laws of computation, the laws of geometry, the laws of mathematics, where do they live, right? So Plato and Pythagoras had this idea that there is a Platonic space of these kinds of things, of these forms. And I think we need to take this seriously, in that I think one thing that evolution is doing is searching through pointers into that space. So I think evolution is not really searching the horrible, complex, rugged space of microstates of all the hardware, the phenotypic space. I think evolution is actually looking for pointers into it's looking for physical embodiments that basically take advantage of this incredible space of forms that already pre-exist. I mean, in a certain sense, it's the physics and the chemistry of the physical world. In another sense, these things, you know, the laws of number theory and all this other stuff, they would be exactly the same if the universe was different, right? If all the laws of physics were changed at the Big Bang, this, I think, and I mean, I'm certainly not an expert on the mathematics of it, but to my understanding, all of those things would still be exactly as true. So there's something else here that isn't captured by our typical accounting of what happens in the physical, you know, kind of the physical world. And for this reason, and so I think that is what evolution is doing. It's finding hardware that is exploiting some of these things, and therefore, and so now we come to the final step, is what happens in novel organisms. Specifically, the reason we make xenobots and anthrobots and various other things, all these synthetic beings, is to explore the latent space around the one point that normal development gives us. So normal development gives us, here's a frog embryo, or here's an oak tree leaf, and this is what it looks like. Here's the shape. Here's that, and we say, okay, we know what the oak genome does. It makes these nice flat green leaves. Fine. Well, turns out that there's a space there's a latent space of possibilities, which which we wouldn't know about if we hadn't seen that there's a bioengineer, a non-human bioengineer, which is a wasp, and these wasps will lay an egg on these leaves, and the signals from the eggs will basically hack the leaf cells to build this incredible, like, red, spiky, yellow looking round thing with these galls, these amazingly shaped galls. We would have no idea that the normal cells, the genetically normal cells of the plant, are able to build this, because all we ever see is one point. We see the normal default behavior. In fact, it's so consistent, it's so robust that we really think that's all it does. But with a little bit of prompting, and I use that word advisedly, with a little bit of prompting, you find out that, oh no, actually there are these other shapes around, and so who knows what else is there. So this is what I think of these synthetic. When I call it, you know, people say, why do you call them xenobots? They're living organisms, or they're animal capsids or something else. The reason I call them xenobots is because I view this as a vehicle for exploring the latent space. I want to see what other forms and functions are around in the space around this what the genomic hardware can do. And so now this is, I think, what is just the barest beginnings of the answer to your question. When we make anthrobots, you know, these are little biobots that are made from tracheal cells of adult human donors. They have all kinds of cool properties. They not only do they form interesting shapes, and they swim around on their own. They're self-motile, but they have new capabilities. Like, for example, they repair neural wounds, and they take neural scratches, and they heal the sides together, and they do some other stuff that nobody's seen yet. It isn't published yet, but hopefully it will be later this year. We'll have some cool stories about them. And so where does all this come from? Or in the case of xenobots, kinematic self-replication, the fact that they can make copies of themselves by coalescing material that they find outside in the environment. I think that what we're seeing here is the ability of biological hardware to explore and take advantage of other regions of this Platonic space of possibilities that allow them to solve problems. And so that's what I think is happening here. That when you say where is it encoded, where does it come from, we have a couple of ingredients to the answer. You have the genomic hardware, which is, well, it's this way because that's where it was encoded. So some of it is certainly genomically encoded. Some of it is emergent in the standard version of a complexity and emergence. You have simple rules like cellular automata, they do stuff, and then boom, there's an. So some of them are hardware encoded, some of them are emergent. Others are really drawing from this from the space of wherever the laws of mathematics and computation come from, right? And others are generated on the fly by the cognitive processes of the system. So these are patterns, much like, you know, where do the patterns in your brain come from? Well, they come from you trying to organize your experience. They come on the fly from you as a sense-making agent. So those are the ingredients, and where do they come from?
Amazing. And so the standard view, because that's sort of also the amazing implications for instance, cancer could be that we now have this paradigm of micromanaging that disease, right? But just targeting it on a hardware level. Whereas if we could do the goal setting on the cellular level, saying to these cells, okay, the goal is again to cooperate, then that could be a future cure of cancer, right? Which could be an implication of your work.
Totally, and we've done this in the frog model. We've already done this. So we've shown that you can you can inject human oncogenes into a tadpole, and they will make tumors. And if you do that, but you force the cells into proper electrical state with communicating with their neighbors, you prevent tumorigenesis, even though the oncogene is blazingly strongly expressed. And that's not because you don't destroy the oncoprotein. You're not managing the cell, you know, the micro level details of transformation. No, what you're doing is you're forcing that cell to stay connected to their neighbors. And when you have a mind meld with a large collective that all says, we are building a nice kidney, we are building a skin, it's really hard for that cell to have an independent thought of, you know what, my goal is to proliferate as much as I can and crawl away wherever life is good, right? Metastasis. So this is, we can actually see this, because the first thing that happens when oncogenes act, first thing they do is they cause electrical disconnection from their neighbors. And as soon as you disconnect it, well, now you're an amoeba again. And as far as you're concerned, the rest of the body is just external environment. So that border between self and world can shrink and grow. Right, so we started both each of our lives, but also evolutionarily, we started our life as a microbe, as a single cell with a tiny little cognitive light cone. We only care about what happens in the cell. Everything else is environment. And with evolution and development, that cognitive light cone grows, and then it could sometimes shrink again. And so this is the, you know, one of the very clear examples of how this stuff is not just you know, wishful philosophical thinking, and you know, no, it has very practical implications that if you follow through, you discover therapeutics.
And I heard you say the word mind meld. I like that a lot, because it's a good thing for an organism in the sense that our cells have a mind meld, which sort of makes that my body functions well. Do you see that possible on higher levels? I mean, what are your thoughts on sort of a Gaia like thoughts on a sort of planetary mind or universal minds? I mean, are you open to ideas like that?
Yeah. So two things I want to say about that. One is that I certainly think it's possible. Absolutely, I think it's possible, but I think it's very important that we don't fall into the trap of, I mean, some people say that's ridiculous, that's a misuse of the terms, and other people say, oh, absolutely, the whole universe is one giant mind, you know, guy. Like, okay, both of those are hypotheses. You cannot decide between those just by sitting in your armchair and deciding which one feels good to you. You need to do experiments, and obviously on the higher level, you know, on the level of the universe, the level of the solar system, I can think of how to do those experiments, but they're not practical. And on the level of ecosystems, you know, maybe it's practical to try some of this stuff. Can you train an ecosystem? Maybe. But again, all of these are empirical questions of how to recognize minds and other embodiments.
Yeah, sorry to interrupt you, because you say you can't think of experiments though not practical, and this is what a lot of physicists do, right? They come up with these. But
yeah.
do you have an idea how you could sort of make that testable from
Sure.
your paradigm? How would that be done?
Sure. So that's one of the things that our framework is trying to do is to develop general roadmaps for discovering novel intelligences. And one of the simplest ways, and this is certainly doesn't go all the way, but one of the simplest ways to know when you've communicated with an intelligence is when you can train it, right? Because even simple minds have certain kinds of learning that they can do: habituation, sensitization, associative learning, prediction, and so on. So you can imagine, like, when somebody says to me, well, your view, then I guess you say even the weather is has a mind. So, well, I don't say that it does. I say that it might. Now the question is, like, could we do an experiment? So for example, you have I don't know, a tornado or some other kind of weather system. Do we could, in theory, if we had the tools to do it, we could in theory test whether they habituate. We could in theory test, can you train a dynamical system? And that might sound crazy. How would you train a hurricane or something? But all of us are just a kind of dynamical system, right? Now we have some qualities that hurricanes and things like that don't have. But again, it's a spectrum. It isn't a categorical difference, and we are all temporary dynamical systems in metabolic space and things like that. And we can learn, and it's entirely possible that if we had a way to stimulate weather patterns in a particular way, you might find that, wow, there really is some kind of habituation here, there's some kind of sensitization here. So you could now a couple of things to say. One is that we've done this with pathways, molecular pathways. This is why I was saying earlier that I don't think cells are the bottom level, because all it takes is half a dozen genes interacting with each other to show associative learning, meaning Pavlovian conditioning. Like, that's it. You don't need the rest of the cell. You don't need anything else. Just from the math alone, for just a few interactive subunits can already give you this. So could weather patterns do it? Could ecosystems do it? I mean, if I had to bet $10 right now, I would say probably they can. But we cannot assume that. We need to do the experiments. And so the experiments are training, communication. Now, right? And so they're perturbative. That's another thing, is you have to do perturbative experiments. You cannot just observe, because when you observe things going, as somebody did these experiments in the 70s where they made these videos of triangles and squares and circles moving around, right? And they would have human volunteers looking at them, and they would make up stories. They would go, oh, it's very clear. The circle is in love with the triangle and the square is trying to push them apart. Like, it's obvious. Okay, so you can't know any of that from just observing. If you want to test the intelligence of your system, you have to perturb it. You have to put barriers between it and its goals. And you have to see how clever is it to pursue it. So
exactly.
You have to interface to the system. So the last and the final thing about that is, it's all well and good when you're testing these kind of primitive systems, these protocognitive systems. But one thing that people often ask is, well, what if we too are part of some sort of giant intelligence, right? So Gaia, so maybe our society or the biosphere or something is this like giant intelligence. Quite possibly. I suspect that it isn't possible to get conclusive evidence of it. I'm going to guess there's some kind of Gödel theorem or something like that around the fact that you really can't know when you are part of a bigger system that's so much smarter than you. I think it's probably impossible to know, but I do think that we can gain evidence of it. And what would that look like, right? What would that evidence look like? So I have a feeling, and again, this is just me going much further than we have rigorous mathematics around. I think what it would look like is synchronicity. I think that what you would see is events that do not look causally connected at your level, but they are connected by meaning. They are connected by some kind of links from the perspective of a cognitive observer. And those kinds of things that don't look like, well, the physics don't look like they should connect together, but they do connect on a meaning level. I think that's what it would look like. If we were part of, we would see patterns in the world, whether they be social patterns, financial patterns, movements of matter and energy in the ecosystem, whatever, that would not have a neat the explanation at the physical level, but they would make sense from some higher level observer's point of view. And so this is like I wrote a little dialogue at one point between two neurons in a brain, and one neuron is kind of like the hard-headed materialist, and he says, look, we live in a cold, unfeeling universe. There's no pattern. Nobody cares about us. It's just, it's just each one of us is alone. And the other one's a little bit mystical, and he says, I don't know. I have this feeling that we're part of some system that actually, the universe actually learns and wants things and does. And the person's, you're crazy. And he said, no, I don't know. There's like these patterns of, of propagate, like back propagating error and whatever. And sometimes, the universe punishes me and whatever. But I feel like there's a purpose behind it. And he says, that's crazy. So right. So from the perspective of in that system, the mystical neuron is correct, because in fact they are part of a larger system. They are part of a larger system that has a that has a global agenda that neither of them could possibly comprehend. But that's it's an experimental thing, I think, to gain evidence for it.
I like your openness and your rigorousness, that sort of you have to sort of avoid sort of going astray with these wild ideas and not making them testable, of course. And there's one sort of deep, I think a bit philosophical question I had. So I read in your AI paper that I liked the meta. You said, and when we raise a child, I have a now 3-year-old, but like 2 years ago, he would like just babble on. I would know he didn't understand the meaning of those words, but he was just trying. And then somehow he came to meaning, right? So you could say it came from syntactical playing around with words to meaning. And then you in your paper say, maybe that could be the same in AI. I was just wondering what your thoughts are here, because sort of the Chinese room experiment and sort of in philosophy, it seems quite well established that there's this great divide, right? This sort of categories of syntax and semantics. What are your thoughts here? Can that be bridged? And do you think, and also, how would you apply that then to your cosmology in the end? Because would that mean that physics has semantics to it? I'm just curious what your ideas here. It really triggered something reading me because you are onto something here. But
I mean, the first thing to say is that what I don't mean is that today's AIs are like human children. So after that piece came out, I got a whole bunch of emails saying, you don't know what children are, you don't know what AI is. Okay, let's be clear. I know what children are. I have to. The modern AIs are absolutely not like human children. Okay, let's fine. But that doesn't mean that it isn't incredibly difficult to specify what is it that magic oomph that we're supposed to have that synthetic or hybrid kinds of agents don't have. I mean, a lot of people feel very strongly that there are machines and then there are organisms. And okay, so what is that, right? What exactly is that? And people have tried to specify this for a long time. I've never seen a good story of it. I think that what we actually have is a kind of scaling. And in terms of syntax and semantics, I think that is a computationalist framing that is useful for some things, but it is not useful for many other things. And I don't believe these are real crisp categories at all. It starts right at the level of, remember the gap junctions, right? The connections between the cells. It's already broke. The distinction between data and machine are already broken there, because what happens is when ions flow through the gap junction, they change the voltage of the other cell, which in turn controls whether the gap junction is open or closed. So what you have here, because the gap junction is also voltage sensitive, it can be. So what you have here is the data, the information that comes through the machine changes the physical state of the machine. That's it. The distinction between pure data and the hardware, that this unchanging Turing machine that just operates on hardware, gone. Biology doesn't respect that at all. So I think semantics is when we say semantics, all we are saying is that there is some observer that is able to squeeze adaptive saliency from whatever it's looking at. So there's a physical set of events, let's say my own memories. I am an agent that knows how to interpret my own memories to some extent, making up a lot of it. I mean, confabulation is not just for language models. Like, we all do it all the time because our memories are very compressed. Our engrams are very compressed representations of the past. We have to constantly reinterpret them, and we add a lot of stuff to it. Real agents and real observers are ones that can interpret and embellish their own memories in real time, and are constantly in this process of.
These are the 3 things I would do if I was starting a YouTube channel in 2025. Number 1.
sense making, storytelling about what they are, what the outside world is, and so on. And so there are multiple observers that can look at the same physical events and see nothing, see something, or see something completely different.
Yeah.
And so no, I don't think there's any
Yeah.
sharp distinction here.
And we spoke about intelligence, and I've listened to a lot of interviews with you, and I know that you're not very keen on sort of questions, philosophical questions like what is consciousness, etc. But if I'd ask you what is consciousness for, and I'm talking about phenomenal consciousness, sorry, what it is for, what are your thoughts here, or your latest ideas around consciousness?
Okay, in the last couple of minutes, I guess the first thing I will say is that I'm not keen on it. I think about this stuff all the time. It's just that I try to spend most of my time, most of my public, work and like my official work on things that I think can make a very practical difference right now. And I don't yet have a theory of consciousness, a new theory of consciousness to offer. I mean, I'm working on something, but it's not useful yet. So I haven't been talking about it. But I will just say this, just two things. One is that I think that for exactly the same reason that we associate consciousness with brains, we should be very open to the idea of consciousness in various other body parts. That's the first thing. And the fact that you are not conscious of it means nothing, because you are also not conscious of my consciousness. You're not conscious of your right hemisphere's consciousness. That doesn't mean anything. But for those same reasons, in terms of intelligent behavior, in terms of all the same mechanisms, all of that stuff is throughout the body. So that's the first thing. The second thing is that what is it for? Well, that goes back to our Platonic space concept, which is, well, what's the fact that NAND is universal, or the fact that the angles of a triangle will add up to, what's that for? So I think we should be really careful in saying that everything is for something from the standard evolutionary point of view. Some things are not necessarily for anything in that selectionist sense. Some things are just features of the universe, and I think that consciousness roughly tracks agency, which we could, we'll need another hour to kind of talk about what I think that is. But basically, the question of what I think consciousness is so Mark Solm says that consciousness is palpated uncertainty about the outside world, and what it feels like to have to make decisions, and what do I do next, and the uncertainty. I think that's I like that a lot, and I think that's very close. I would say that consciousness is the palpated uncertainty about what your own memories mean. So it's a constant real time. It doesn't feel like that. It's not how we describe it. But I think what it really is under the hood is our constant sense making. This idea that as an agent, it's not just about looking backwards and explaining what happened. It's about looking forwards and saying, what is the best story I can tell about myself in the outside world that helps me know what do I do next in this uncertain, dangerous world. And so that is what I think consciousness is. And I think we have there are many open questions about why it feels certain ways and whatever, but fundamentally that is what it's for, because without that process, I don't think anything could survive. But also I think it's an important fundamental property of the universe that you cannot tell a simple selectionist story about it.
Michael, thanks so much. And I just have one, and maybe you can answer short. And I think you've already given an answer with this last answer you gave me. But I thought just the final question for you: how has your work changed your personal life? I mean, your outlook on life. I just heard you say that you're a father of two, because I can imagine working on stuff like this, it does something to how you just live your life.
Yeah, I think it does, and I think it has serious, and I think we need a lot more time to talk about this, but I think it has real implications for ethics and for how you relate to other beings, and how you think about enlarging your sort of light cone of compassion and things like that. And so yes, I definitely have some thoughts about it, but I think we'll need a different.
It's for our next time. Thanks so much for your time. And
Let's tune in for that one.
Okay, so there you have it. That's a snapshot of what Michael Levin is looking at. Let me just go. I think this is where we're at here. Let me go back here.
He's like
Yes.
an example of, if you fast forward the evolution of Darcy Thompson, he might end up here,
Yeah, and
like Michael Levin.
but you have a huge he has a huge knowledge of the computers and everything that goes with that didn't exist when
Right.
yeah.
Well, that's
So
what I
anyway,
mean, but
yeah.
it's like a foreshortened, you know,
Right.
you wouldn't think that would happen in 100 years.
No. Hold on a second. Let's see if I can get us into. I've got to get us back to. If I go here. It's a PowerPoint slideshow. Oh, I see what the problem is. Let's go here. I'm going to go back to the start here and skip. Okay. So this is an example of a TED Talk that Michael gave in 2021 that I encourage you to look at. It's only 20 minutes long, but it gives you some idea of how he's coming about this stuff. It is dated, of course.
I don't think his work would be at all possible without a similar multidisciplinary approach like Thompson's.
Right.
You know, it's not just one lens that he's looking through.
No.
So that's why it ends up so multifaceted and multidimensional, because he's looking through multiple lenses at every detail that he's looking at, and then he's putting it together in a picture that you couldn't possibly form. It's very synergistic, you know,
Right.
the way it comes together.
It's also very different than the bottom-up reductionist view that if I just start with the DNA and the chemistry, biochemistry that's here, I can figure it all out.
And that kind of mechanistic view.
Yeah, and the complexity of that is hard to appreciate how, you know, all of these things. I've already said we have 50 trillion cells in your body, each of us. So go figure out, and each of those cells, as Michael said, is by itself is bordering on conscious at some sense. So something like this, it gives you a chance to try and figure out what really is possible. But the whole point is unification here. He's looking for things that can scale up. And he's drawing on taking cells and groups of cells, but they have collective intelligence, and then coming up with things that go with it. So
But then it's those collections of cells that are interpreting their own experience.
right, and they're going toward goals. So some want to become a brain, some want to become a muscle, some want to become a skin, some want to become a bone, and on and on. Whatever organ of the body you want to talk about. And so it's the intersection of all these things. And the thing that we're dealing with there is spatial information, which is with everything going on, but also temporal information that is letting these things occur. And so you have to keep track of all these things in order to figure out which end is up. And this is where he's looking for really the fundamental nature of real living creatures, you know, and he tests this by life as it could be by trying to build these artificial life entities, which he calls xenobots. And this is as much based on information or computer science, or viewing this as a branch of mathematics as it is biology. So
I loved it when he said that what would it look like, that kind of communication between these seemingly disparate, and he said synchronicity.
Right.
That was my favorite moment of the whole thing.
No.
And this reminded me of a poem that I wrote decades ago. I'd like to dig it out now, about humanity as like a virus or a cancer on the back of Gaia, you know, that because we are kind of running amok from the whole, from nature, and making things, we're making these other, subset things that are act as cancerous agents or cancer causing agents on the whole. That we're not in harmony with
Gaia.
with Gaia.
No, not at all.
With each other, or even ourselves,
Right.
or much less with Gaia. And is that something wrong in the program? Are we, a blip in the program?
Or if Gaia's intelligent, why wouldn't Gaia simply shrug? And that'll take care of killing off a lot of this disturbances that Gaia senses.
To shrug us off her back.
Yeah. So he's clearly looking at very much top-down mechanisms and trying to take advantage of what's already there, with bioelectricity being one of the things. So it covers molecular genetics, biophysics, and then a lot of computational modeling. Really, you couldn't do this without modern computers and software. He's looking at regeneration, embryos. Here you have in embryos, you start off with an egg and a sperm, and the cells divide. When you get up to 100 or 200 cells, all of a sudden the cells start to differentiate themselves, and some become bone cells, and some become neurons, and some become skin. And where in the world is that in all of this? There's something at a certain level of mass that it processes all these things and knows what to do.
And they seem to be identical up until that point.
Yeah, no one can. It just magically changes. It's like throwing a switch. The bioelectrical signals seem to be the way that they communicate. And he talked about here the use of these basic patterns that are in some higher dimensional space that these things are following. So the pattern says, I'm going to become the finger on a hand. I'm going to become a hip bone. I'm going to become the right hemisphere of your brain, and on and on. Where do these patterns, where do they, how do they do this? And so a lot of this is looking at how to deal with diseases of a different sort and regeneration, but in a very different way than you'd see with conventional pharma. And this is in many ways still basic research, because the questions that are being asked here are really fundamental and foundational in all of this. It's meant to complement the mainstream, the reductive analysis, with the synthesis going top down to control everything. And there's a lot of use of artificial intelligence and neuroscience that he just touched on to try and figure out what's going on here. Very much algorithmic, lots of things dealing with shapes, morphogenesis, and so forth. Where do these specs come from? They come out of nature somehow, out of a Platonic space that Plato and Pythagoras sensed. And here's a little graphic he has showing this biological decision-making that leads to some complex, novel architectures using AI, which leads to this AI is very much machine learning, which in turn leads to uncovering algorithms. Now the algorithms feed back into the biology, so you run around and around here doing all this. And the thing that intelligence is collective, he already said that he thinks cells are at some level of intelligence. So every cell in your body is intelligent at some level, but collectively you end up with something bigger than you ever had.
And that it's very holographic.
Right.
I thought that was interesting.
Yeah. So now we're going to leave him and go to another person at just equal time, Rupert Sheldrake, who has been around for a while, and he has what he called a theory of morphic resonance, the natural systems inherit a collective memory from previous things of their own kind. And this influences them through what he called the non-physical morphic field. And so they're natural laws that build up over time. And you end up with new compounds that crystallize more easily after they've been, you tried to synthesize them repeatedly. It becomes easier and easier. Or why learning a skill becomes easier with practice. Or the one that I like, when you stare at someone in a crowd, all of a sudden they'll sense that and turn and look at you. And you can go try this yourself, and you'll see, I think, that it's all too common. Well, what in the world is going on there? Are your eyes doing something, sending out some photons that somehow the person you're looking at senses? I don't know, but it's something that I've seen repeatedly. Now he gets attacked because these are, a lot of hand waving here, and what experiments could you really run? But he says that nature really deals with habits and isn't governed by fixed laws. How could it be easier to form a crystal if you've done it again and again? So somehow nature iterates and learns this so it can reach that goal more quickly after you've done it enough times. And he talks about this collective memory, and then finally this morphic field, some sort of non-physical field that carry information that are responsible for shaping the form of an organism. Maybe there is such a thing. And so you have memory, and then he has examples of this, why dogs often seem to know when their owners are coming home. All of a sudden, the dog will get up and start barking or going to the door. I already mentioned staring at someone, and they'll turn and stare back at you. Why would they do that? Learning a new skill becomes easier with practice, and so forth. But as you can see, his program here it's interesting, and it's thought-provoking, but by no stretch of the imagination is it the program that Michael Levin and his grad students are pursuing. And so we'll see what happens. There's also this so-called 100th monkey phenomenon. There was a young macaque on the island of Koshima in Japan that began washing sweet potatoes. And over a period of time, other monkeys on the island started doing this. And once there was a certain critical mass, supposedly 100 monkeys, all of a sudden, all the macaques on all the nearby islands, including the mainland of Japan, started washing sweet potatoes, even though there wasn't any direct contact. So there's something going on here that we don't know. A different example, Swathing, England in 1921, there were some birds that came by. There used to be milk delivered to everyone's doorstep with a tinfoil cover, and the birds, this is a blue tit, would poke through the tinfoil and grab the cream. And this was pretty good stuff, getting cream. And so after a while, more and more birds in this area started doing this, even though, where did they learn it from? And then pretty soon, all the birds all over the place were doing it, which led to them repackaging this tinfoil.
The humans learned.
Yeah, and so forth. So really where we're going here, we go to Star Trek out of the 1960s. Here's our Starship Enterprise here, and here's what's called a med bed. And in this med bed, you would sit down and lay in it, and your body would resonate with this morphic field that's here, that's being created by this, and it would resonate toward what is a healthy body. So what isn't healthy in this collective intelligence would simply die off very quickly. You would cure yourself, you'd regenerate limbs, you'd wherever you're cut, it would get healed very quickly, and so forth. And what also comes with this is the ability to identify root cause. for what in the world the problem is. If you remember in Star Trek, the medical officer had a scanner and simply scanned you and said, "Oh, you have this," immediately. None of this getting out a stethoscope and listen to your lungs. There was the scanner that magically did all this stuff. So this is the goal. We're not here today, but it's the thought that goes with it. So with that, we're done. Thank you for listening. Marjorie, anything else?
Well, thank you for coming back, and we hope that this follows on the heels of part 1 in a way that makes sense to you. We're finding this very exciting and affirming of where our intuition has been leading us for quite some time. We've both been thinking about these things from different points of view for decades, and it's nice for us to come together on this common ground and see it from all these different points of view, because we think this is a view of, you could call it the collective unconscious, or you could see it as the divine. It's not necessarily, a puppet maker, working with a hammer and a chisel and a slide rule and a protractor, but it's how it works. It seems to be a view of how evolution happens and just the whole phenomenon of life. And I love all the signs of the interconnectedness, but there's also so much differentiation, and where are we on that scale? If it's a question of scale, do cells or cell nuclei have consciousness? We don't really know. Do atoms have consciousness? Does the planet have a consciousness, or the solar system, or the sun, or the galaxy? And where are we on that scale is I think it's an endless meditation, at least. And just a rich contemplation to engage indefinitely.
Well, on that note, we will close, and we look forward to further pursuing this. Have a good, bye-bye.
Till next time.